In this video, we will look at the general senses. Let's start by defining different types of receptors. The group of exteroceptors monitor your external environment. Examples of exteroceptors that would monitor a stimulus outside the body include the photoreceptors in your eye, noticing changes in light, pain receptors from a cut, or for example this picture, a sharp pin poking on you. If somebody were to touch you, like tickling or pinching, or wind and rain, anything coming from the outside environment that is being monitored by your body is done so with exteroceptors. Therefore, interoceptors are monitoring the inside environment, so all the stimuli coming from your internal organs. And a term for organs is viscera, so these are also known as visceroceptors. For example, a full bladder, the blood pressure being high in your blood vessels, pain coming from inside the body. And there's a type of special interoceptor, a class of its own, called proprioceptors. Proprioceptors are receptors embedded in your muscles and tendons that monitor your body position, and they are continually sending messages up to your brain, especially the cerebellum, so you can balance your body and coordinate your movements. We will look at a couple of exteroceptors called Meissner's corpuscles and Pacinian corpuscles. Meissner's corpuscles will be involved in light touch, and Pacinian corpuscles will be involved in deep pressure. Therefore, if you look at their location, in order to be involved in light touch, a Meissner corpuscle has to be closer to the surface of the skin. And deep pressure, where you're really pushing on the skin, that's going to be found deeper. So let's remind you of the layers briefly. The top layer of the skin here is the epidermis, and then the thicker part of the skin we call the dermis. But remember, there were two parts to the dermis. There were this upper thin layer we call the papillary layer, and that's where the Meissner corpuscles are found. The Meissner corpuscles are in the papillae, the raised projections of the dermis, and they're just below that epidermis, very close to the surface. If you look at the histology here, they look like a swirling of cells, almost like the tip of a Q-tip. But further down and much larger is the Pacinian corpuscle. This is the one for deep pressure. You really have to push on it to activate this receptor. And notice how it's located deeper down in the bottom of that reticular layer. And under the microscope, it has much more encapsulated, bulbous look to it. One of the things we will test and look at in the lab is receptor density. So how many receptors are located in an area on the body? You have areas that have more receptors packed into a small area, and areas where the receptors are more spread out. And this is what we call a homunculus, which is just a figure that shows you how much brain power is devoted to certain areas of the body. And you'll notice it has some exaggerated parts. Look how big the face and the hand are compared to the leg. The leg doesn't have hardly any area on the brain. You have a lot more brain power devoted to areas where there is a higher receptor density. In other words, the more receptors, the more stimulus that is coming in, the more incoming information the brain is getting. So there will be a larger amount of brain power devoted to those receptors. And so the areas where we have a higher density, that means more cells packed into a smaller area, will have more brain power. We will do some lab demonstrations to illustrate the density of receptors. And the first demonstration we'll do is called two-point discrimination. For each of these, you should understand the equipment being used, how the test is performed, and what the results indicate. In the two-point discrimination, we will use calipers, where you take this pointy edges and you slowly pull them apart and tap them on your lab partner's skin until they tell you it feels like two different points. Once they tell you it feels like two different points, you will record the measurement and you will look to see which parts of the body have small measurements, what we call a small two-point threshold, or large two-point thresholds. The smaller the measurement is, the more the receptors are packed in there and the higher the density. In tactile localization, you will use different colored markers. This is going to test your ability to reproduce where you're touched without looking. You will not use any visual help. So while your eyes are closed or your head is turned, your lab partner will touch a marker to your skin. And then you will try to touch that same area with your marker. In an area where you have more receptors and a higher density, you'll do a better job of hitting that exact same spot. In an area where the receptors are further spread out, you will have a lot more distance between the two colored markings. Adaptation is where your receptor gets used to a stimulus and quits sending signals to the central nervous system about changes. Receptors send signals when there is a change in the environment. But as that change stays steady and it's no longer fluctuating, then they reduce the number of signals that are being sent to the brain. For example, if you get into a hot tub, initially it feels really hot, but after you've been in it a few minutes, it doesn't feel as hot, even though the temperature's still the same. When you first got in, that was a huge change in temperature. But after you've been there and the temperature's been that same constant heat, you start to send less signals to the brain. We're usually familiar with this with smells as well. You walk in and something smells stinky, and then after a few minutes it doesn't smell as bad. So your receptors are adapting. They're getting used to the stimulus. But not all receptors in the body adapt. For example, your pain receptors, what we call nociceptors, they do not get used to pain. Pain means there's damage in the body. And so your receptors for pain will always send signals for pain because you don't want to get used to damaging the body. Referred pain is where the body will misinterpret the signal and you will feel sensations somewhere else than where it's actually originating. So for example, if you've ever hit your funny bone, you hit the elbow and that's where the stimulus is. But you can feel it travel all the way up to your fingertips and tingly sensations, even though you didn't touch your fingertips. If you look at what we call a referred pain map, if you have right shoulder pain, you see here it's indicated in purple, that could indicate gallstones. You may be having a heart attack, but yet feel pain in your left arm. Referred pain is the brain's misinterpretation of the information. A lot of the signals travel through the same tracks, the same pathways, and so initially the brain may interpret the signal as originating somewhere else. But eventually it will work it out. For example, that heart attack, after a while, the pain in the left arm will actually turn into chest pains, and it will correct itself. For this demonstration, we will use a bowl of ice water, and we will demonstrate in lab what happens if we place our elbow in ice water for 5 minutes. And that concludes general senses.